Every organism carries out various life processes. All these processes are collectively called physiological processes. Constant supply of energy is essential to carry out these processes. Living organisms derive this energy by oxidation of food. Only green plants and cyanobacteria can synthesize their own food by the process of photosynthesis. Even in green plants, only those organs which possess chloroplast perform photosynthesis, but food is required for the oxidation by all the organs, even which do not possess chloroplast. So, synthesized food is translocated to all non green parts. Most important part is that all the food that required for life processes comes from photosynthesis. Thus, energy exchange takes place during biological processes. Photosynthesis is an endergonic process where respiration is an exergonic process. The breakdown of carbon-carbon bonds of complex compounds through oxidation within the cells, releasing considerable amount of energy, is called respiration. It takes place in all cells, so it is also known as cellular respiration. The substances which are oxidized during this process are called as respiratory substrates like carbohydrates, proteins, organic acids, and fats. Both plants and animals respire. Respiration is an important physiological process taking place in the mitochondria of all the cells of all organisms. A part of the energy released during this process is utilized for the synthesis of ATP. The organic nutrients synthesized by the organisms or taken in as food undergo slow combustion during this process. Plants require oxygen for respiration to occur, and they also give out carbon dioxide. But plants, unlike animals, they do not have specialized respiratory organs. Stomata and lenticels are present, which are actually taking part in gaseous exchange. There are various reasons why plants do not have specialized respiratory organs. Each plant part is directly involved in the exchange of gases. As plant parts respire at rates far lower than the animals do, the need of gas exchange is not much. When cells photosynthesize, availability of oxygen is not a problem in these cells since oxygen is released within the cells. The strategy that the plant cells use to catabolize glucose molecule in such a way that not all the liberated energy goes out as heat. During respiration, the stored energy in glucose is gradually released through controlled oxidation. The released energy is stored by forming ATP molecules from ADP. During the process of respiration, oxygen is utilized and carbon dioxide, water and energy are released as products. The combustion reaction requires oxygen, but some cells live where oxygen may not be present or may not be available. Among present-day living organisms, several of them are adapted to anaerobic conditions. Some of these organisms are facultative anaerobes, but some others, the requirement for anaerobic condition is obligate. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is called glycolysis. Glycolysis is common to all living organisms. Breakdown of glucose to pyruvic acid is called as glycolysis. The term glycolysis has originated from the Greek words. Glykos means sugar and lysis means splitting. The scheme of glycolysis was discovered by Gustav Emden, Otto Meyerhof and J. Parnas, and the glycolysis is known as EMP pathway. Both aerobic as well as anaerobic respiration begins with glycolysis. In this phase, oxygen is not utilized, and glycolysis occurs in the cytoplasm of the cells. During glycolysis, glucose is converted into 2 molecules of pyruvic acid. In plants, glucose is synthesized during photosynthesis and then it is converted into sucrose. The enzyme invertase converts sucrose into glucose and fructose, which readily enter the glycolytic pathway. In the first phase, the molecule of glucose is phosphorylated to glucose 6-phosphate with the use of ATP. The phosphate liberated from ATP is joined to carbon number 6 of glucose. ATP becomes ADP and the enzyme responsible for this reaction is hexokinase. Then glucose 6-phosphate is converted into its isomer fructose 6-phosphate through molecular rearrangement. Now this fructose 6-phosphate is again phosphorylated with the help of ATP, forming fructose 1,6-biphosphate. And during this reaction, ATP is converted to ADP. Now this fructose 1,6-diphosphate splits and 2 3-carbon molecules are formed, the phosphoglyceraldehyde and dihydroxyacetone phosphate. 2 molecules are formed, phosphoglyceraldehyde and dihydroxyacetone phosphate. Now both these 3 carbon sugars are interconvertible, and dihydroxyacetone phosphate is converted into phosphoglyceraldehyde. So, 2 molecules of 3-phosphoglyceraldehyde are formed. Energy will now be released in the subsequent phases. Till now, 2 molecules of ATP are consumed. Each molecule of this 3-phosphoglyceraldehyde is oxidized, and 2 H plus and 2 electrons are released from it. The released components are accepted by NAD, forming NADH and H plus. Simultaneously, a molecule of inorganic phosphate is also added. As this happens, 1,3-biphosphoglyceric acid is formed. This phase of reactions constitutes phosphorylation phase. Next is the phase of dephosphorylation. 1,3-biphosphoglyceric acid is dephosphorylated and is getting converted to 3-phosphoglyceric acid. The phosphate formed from the substrate combines with ADP and forms ATP. This is called substrate phosphorylation. Now this 3-phosphoglyceric acid is first converted into 2-phosphoglyceric acid and then into phosphoenol pyruvic acid. Water is released during this process. Phosphoenol pyruvic acid is dephosphorylated and converted into pyruvic acid. The phosphate released from the substrate combines with ADP and forms ATP. Thus, at the end of the process, 2 molecules of pyruvic acid are formed from 1 molecule of glucose. 2 molecules of ATP are consumed and 4 molecules of ATP are formed. So there is a net gain of 2 ATP molecules. 2 molecules of NADH2 are also formed. Now there are 3 major ways in which different cells utilize pyruvic acid produced during glycolysis. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. When respiration takes place in the absence of molecular oxygen, it is said to be anaerobic respiration, and this results in the breakdown of glucose incompletely. So incomplete breakdown of glucose takes place in anaerobic respiration. During this process, carbon dioxide and organic compounds like ethyl alcohol or lactic acid are produced and some energy is released. Water molecule is not produced during anaerobic respiration. So these reactions are called fermentation. The fermentations are of 2 types: alcoholic fermentation and lactic acid fermentation. First we will see about lactic acid fermentation. In this type of fermentation, the pyruvic acid is converted into lactic acid by the enzyme lactate dehydrogenase and coenzyme NADH2. The NADH2 is oxidized to NAD. Lactic acid fermentation takes place in the muscles of animals during exercise when oxygen is inadequate for cellular respiration. In alcoholic fermentation, this occurs in the yeast. The incomplete oxidation of glucose is happening under anaerobic conditions, where the pyruvic acid is converted to carbon dioxide and ethanol. The enzyme pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. The pyruvic acid is first decarboxylated to acetaldehyde in the presence of enzyme pyruvic acid decarboxylase and thymine pyrophosphate. Acetaldehyde in the presence of the enzyme acetaldehyde alcohol dehydrogenase and coenzyme NADH2 is reduced to ethyl alcohol. NADH2 is oxidized to NAD. So ethyl alcohol is formed in alcoholic fermentation. In both alcoholic and lactic acid fermentation, less than 7% of the energy in glucose is released, and not all of it is trapped as high energy bonds of ATP. The end product is either lactic acid or alcohol. Is oxidized to NAD. So ethyl alcohol is formed in alcoholic fermentation. In both alcoholic and lactic acid fermentation, less than 7% of the energy in glucose is released, and not all of it is trapped as high energy bonds of ATP. The end product is either lactic acid or alcohol. Is oxidized to NAD. So ethyl alcohol is formed in alcoholic fermentation. In both alcoholic and lactic acid fermentation, less than 7% of the energy in glucose is released, and not all of it is trapped as high energy bonds of ATP. The end product is either lactic acid or alcohol.